On behalf of the NIH Director, it's my pleasure to introduce today's NIH Director's seminar speaker, Craig Thompson. Dr. Thompson, as you all know, is President and Chief Executive Officer of Memorial Sloan-Kettering Cancer Center, where he's been for the last couple of years. His background includes a bachelor's degree from Dartmouth College and an MD from Dartmouth and the University of Pennsylvania. He did a residency in medicine at the Peter Bent Brigham Hospital and University Hospital in Boston, and a hematology-oncology fellowship at Fred Hutchinson Cancer Research Institute, University of Washington. And one theme that you'll see in Dr. Thompson's bio is he's moved around quite a lot, which is interesting and has been very productive for him. After Fred Hutch experience, he came to this area and actually worked for several years at the National Naval Medical Center as a physician and scientist. After that experience, he went to the University of Michigan, where he became an assistant professor, moved up through the ranks, became an HHMI investigator, and also had a joint appointment at the University of Chicago, where he was director of the Gwen Knapp Center for Lupus and Immunology Research. And it turns out that when Dr. Thompson was at the University of Michigan, he shared a floor with two of our NIH dignitaries, Gary Nabel and Francis Collins. They were all assistant professors together, and you can imagine with the other group that were there what a powerful intellectual force they must have been at that university. In 1999, he came to the University of Pennsylvania, where he became professor of medicine and initially scientific director of the Leonard and Madeline Abramson Family Cancer Research Institute, chairman of the Department of Cancer, and ultimately director of the Abramson Cancer Center at the University of Pennsylvania. And as I mentioned, a couple of years ago, he moved to Memorial Sloan-Kettering, where he leads an incredible program of cancer research and treatment. So I think all of these positions reflect the enormous ability to galvanize exciting new discoveries in cancer research. Dr. Thompson has contributed in, I think, three major areas. The first, which is the work he started, I think, at the Navy Center here and also at the University of Michigan, was to explore the role of oncogenes in hematopoietic cancer and also in lymphoid development. Many contributions there. And then he worked on mechanisms of apoptosis and their role in cancer, and you're probably familiar with a lot of his contributions there. And over about the last decade, he's really pioneered a new understanding of the way cell metabolism interacts with issues related to cell growth and the development of cancer. And some of the work that he's done will be discussed today. In addition to his many significant positions as a scientist, his work has been recognized by election to the National Academy of Sciences, the Institute of Medicine, American Academy of Arts and Sciences, among others. And he's had quite a few awards, and I won't go through them all. He's associate editor, for those of you who are interested, of Cell, Immunity, and Cancer Cell, are some of our favorite journals. He's a board member for many significant organizations and a member of the Lasker Prize jury. So we really have to be nice to Craig. In addition, he started in his career 3 biotech companies, but his most important contribution is that for many years he was chairman of the Board of Scientific Counselors of the NCI Basic Science Division, and many of you who are cancer researchers got to know him in that capacity. Well, I think we should welcome Craig Thompson, whose talk today is entitled IDH Mutations, Oncometabolite Deregulation of Epigenetic Remodeling. Craig.
Thanks, Mike. Thanks. Well, it's a privilege to be back here in Mazer Auditorium and get a chance to give this talk and see so many old friends from NIH days and in science. I have said this before, but I am always embarrassed when we give that kind of introduction because, as my mother tells me when she's occasionally comes and listens in an audience like that, it really sounds like you can't hold a job. What I am going to talk today is to give sort of an overview, since I think it's appropriate on these Wednesday afternoon conferences to sort of, for the students and postdocs, to give you an idea of how we got into this field of metabolism. And I put metabolism only briefly in the title as an oncometabolite, and we will cover that at the end. But I wanted for the students to sort of get an understanding that no one these days, other than kicking and screaming, gets dragged back into the field that we all got taught, which is biochemistry as an undergraduate or metabolism in its latest forms going forward. The people that have done the work that I'm going to talk about today in my lab currently are here. A number of postdocs have led to have their own laboratories in this field of cancer metabolism. But most recently, some of the new work I'm going to talk about has been a delightful collaboration with Ross Levine at Memorial Sloan-Kettering and Ari Melnick around our hematopoietic malignancies. And what's not shown here, but more recently just been published with Tim Chan and Ingo Mellenhoff on gliomas. We've had a long-term collaboration with Lou Cantley and Josh Rabinowitz to bring new approaches to metabolic studies. And a few years ago with Lou and TAC-MAC, I started a company called Agios Pharmaceuticals, which has been looking into the space of cancer metabolism to see if there are any therapeutic ways to intervene. And they've contributed to some of the basic science I'm going to talk about today. So that's the introduction and the people that did this. As I said, we were all, everyone in this room, it's sort of interesting, we spent a significant period of time in science, but every single one of us shares a common background in that we took exactly the same biochemistry course. And if you took it multiple times in high school and college and then in graduate school, you took exactly the same course over again multiple times, maybe in a little more depth. And that's because there hasn't been a new metabolic pathway introduced into a biochemistry textbook since 1983. And then it was only after 10 years of arguing they added one step I will talk about briefly. Before that, the previous new introductory step was in the 1960s. What we learned in our first introduction to the biochemistry of life was that in fully differentiated tissues extracted nutrients from their environment. The preferred nutrient that we share with all cells in our body is glucose. That's metabolized to the hexose sugar, to the triose sugar pyruvate, and then fully burned to CO2 and water in the formation of 36 ATP equivalents on average, to maximally extract the energy in the carbon-carbon backbone of this organic molecule. And that allows cells to produce enough energy to maintain their homeostasis. Particularly 50% of all energy in cells is to pump out the ions that actually diffuse into a cell, sodium, sequester calcium, et cetera, allows the cell to survive another day to survive. We learned in a more advanced way of thinking about organismal biology that in fact in the absence of oxygen, cells can survive for brief periods of time purely through glycolytic metabolism that ends in the product lactate, but that is highly inefficient if the goal is to derive maximum ATP production and therefore survival, because it only requires 2 moles of ATP for every mole of glucose. Now, on an organismal basis, that's not a disaster because, as the Coreys demonstrated, a husband-wife team working in a cancer center in 1947, for which they won the Nobel Prize, that if you recycle this lactate on an organismal basis, you only give up 2 of the ATP equivalents if you subsequently do gluconeogenesis from this lactate and then recover it into oxidative metabolism. And that's what we were told was the way in which cells dealt with their energy demands. And that the whole system was homeostatic, so once you learned it and you passed the test, you could go on to the cool stuff. You could study cell biology, you could study molecular biology, more recently you can study structural biology. But there's been a little nagging problem about that, and that is that when people studied highly proliferative tissue, and the best recent example is if we study the biology of ES cells or iPS cells and maintain them optimally in culture, they don't adopt this metabolism. In fact, what they do is they scavenge all the glucose out of the environment, and even in the presence of molecular oxygen, preferentially throw the vast majority of the carbon that they take up away as lactate. That was actually first noticed in tumor cells back in the 1920s by the pioneering biochemist who contributed to much of this information, Otto Warburg. And so the preferential use of glucose metabolism through glycolysis has been termed the Warburg effect, or aerobic glycolysis when it occurs in the presence of molecular oxygen. And for over now 80 years, scientists every decade have tried to figure out why it is that tumor cells, and now more recently proliferating normal cells, actually adopt this abnormal metabolism. My interest in this did not stem from any interest in metabolism. It came from what Mike talked about. In our studies to understand lymphocyte proliferation in the mid-1990s, we came to appreciate that, in fact, every one of us goes through huge clonal expansions of lymphocytes as we encounter new bacteria or new viruses in our environment and have to fight them off by a specific immune response. And that by this point in our lifetimes, you've had hundreds of those encounters with foreign pathogens. And if you'd kept all the lymphocytes that you created to protect you during those episodic events of infection, you would actually need a wheelbarrow to carry the extra cells along with you to maintain homeostasis. And so other investigators thinking about that problem in every lineage of the body came up with the idea that there must be mechanisms that deleted cells from the body, and the simplest mechanism to delete a cell from the body would be an organized form of cell death, which became known as apoptosis, first with its genetic discovery in the worm. A few years ago, to try and explain how it is that we keep track of every cell in the human body, every one of you walked in here with 60 trillion cells, and how you keep track of 60 trillion cells when actually any one of them could die by apoptosis in the next 15 minutes became the puzzle of the 1990s for the people that are interested in the field of cell death. In fact, that was simplified by Martin Raff in the mid-1990s with the statement that, in fact, he was pretty sure that organismal homeostasis is maintained because every cell in your body wakes up every morning thinking about suicide and has to be talked out of it by nearest neighbors to maintain homeostasis. And so the model simply said that single cells taken from our body, placed in isolation without the normal nearest neighbor signals from their environment, actually initiate over a defined period of time this apoptotic cascade that effectively and silently eliminates them from the body, so-called death by neglect. And the argument became that you needed specific instruction signals to maintain the viability of that cell over the next few years. Those were first known as survival signals, and as I'm going to show you in a minute, in the late 1990s, we proposed that the commonality of those signals is that they instructed cells to take up sufficient glucose to maintain ATP production and homeostasis. Secondarily, though, we also know to maintain organelle homeostasis, to maintain the proliferative advances of lymphocytes, you need additional signals, and those signals harness not nutrients, but the new transcription and translation that's necessary for a cell to enter and progress through the cell cycle. And if this model is correct, it suggests, as I'll show you again in a minute, that there are actually two fundamental barriers to the regulation of cell survival and ultimately cell autonomous proliferation. The cell needs to acquire the information code to proliferate, and it needs the nutrients to enact the survival during that proliferation response. And so, starting with that hypothesis, Jeff Rathmell in the laboratory in 1999, he's now down at Duke carrying out this work in his own laboratory, started to look at individual cell types for death by neglect and to characterize what actually happens. And so the simple experiment is you take your favorite cell type out of the body, you make a single cell suspension, and then you simply ask what happens to the cell during the next 2 days before it commits irreversibly to apoptosis. And what he found in every cell type that he was able to examine, in the absence of serum-derived growth factors, in the first 12 hours, every cell processes the nutrient transporters necessary for taking up nutrients from the extracellular environment off of the cell surface and delivers them to the lysosome, and where they're completely degraded. At that point, the cell starts a cell clock. That clock is dependent on the cell type and the number of intermediate metabolites the cell has available to it intracellularly. But ultimately, that cell over the next 24 hours will have a declining bioenergetic signature read out at multiple organelles that activate so-called BH3 proteins that act ultimately at the surface of the mitochondrial membrane to activate the death-inducing properties of the proapoptotic members of the BCL2 family, BAX and BAC. And through this process causes dissolution of the outer mitochondrial membrane and the initiation of this orderly form of cell death known as apoptosis. So we proposed, as I just suggested a minute ago, that the key factor that regulates the survival is actually the loss of the ability to take up nutrients and therefore to maintain bioenergetics. But there was an opposing view, and that opposing view is That, well, you're not being stimulated by the growth factors in your environment any longer. You can take a more indolent biology, and that biology just simply allows you, at the level you can detect them, to see the decline in nutrient transporters. They're still there in adequate quantities to maintain the viability of a cell. And so we tried to address that question by creating with Stan Korsmeyer's laboratory a mouse completely deficient of backs and backs, so it couldn't initiate apoptotic cell death. And then simply asked, in cell lines and in the whole organism, what happens to cell survival when the cell still has experienced neglect, loss of extracellular signaling, but no longer can induce apoptosis? What we discovered about 6 years ago was, in fact, that cell now turns to a more ancient survival pathway that was previously well described in single-cell eukaryotes and plants, the process of macroautophagy, in which the cell sequesters out parts of its intracellular cytoplasm and sequesters them in a vesicle known as an autophagosome, and then delivers that vesicle's contents to the lysosome for pH-dependent degradation of the proteins and lipids back to the elemental constituents to provide substrates for the mitochondria to maintain bioenergetics. And that allows cells to survive for another 2 to 4 weeks, when ultimately, because self-eating is not a long-term survival strategy, ultimately you run out of things that you can eat from your intracellular contents, and the cell dies necrotically. And so the critical lesion that regulates cell survival we have been working on for the last 12 years is the lack of the cell-autonomous ability of metazoan cells to take up nutrients from their environment in the absence of instructive signals from growth factors, hormones, or other paracrine signals. And so just to demonstrate that, this is an animal that has no apoptosis. Now, it's had a lot of developmental problems, but it's reached adulthood. It's a perfectly happy mouse. It runs around its cage all night looking for food. It sleeps all days with its brothers and sisters. It maintains lineage homeostasis in every major organ system. Now, every major organ is slightly perturbed because of its developmental problems in getting there, but it's able to be a fully functional mouse in the absence of apoptosis. It doesn't become greatly distorted. And since this is meant to be a cancer ceremony, the interesting thing is we had predicted, based on the original models of apoptosis, that this animal would be highly tumor prone. We have kept these animals alive now for over 10 years. We have never seen a spontaneous tumor in these animals. You breed in an oncogene and you will see a tremendous advancement of the oncogenicity of that oncogene in the absence of apoptosis. But we have never seen the animals develop spontaneous tumors. So that led fundamentally to a different change in at least the way we were thinking about things. Instead of the paradigm that I was taught as a student, that there's a fundamental barrier that all cells have to becoming spontaneously transformed into human, which is the loss of the ability to proliferate in a cell-autonomous fashion, and that's because you need growth factor signaling that instructs new transcription and translation. There's a second barrier that exists to cell transformation, because even if one acquires the cell-autonomous ability to take transcription and translation that allows the cell to enter and progress through the cell cycle, this signaling and transcription and translation are highly energy dependent, and they'll deplete the cell's intracellular metabolites just as quickly as if we'd eliminated the nutrient transporters in the absence of a compensatory increase in their ability to take up nutrients. And so this would be pro-apoptotic in the absence of a concomitant signal, a second barrier, in which the cell must be instructed to take up adequate nutrients from the environment to maintain ATP production and also to maintain macromolecular synthesis, since we both need nutrients to support bioenergetics, but also to support replacement biosynthesis of proteins and lipids. And if you think about that from a cancer cell's point of view, it has a really interesting set of predictions that allow you to test things. About the time we were thinking about this, people were investigating various oncogenic forms of signal transduction adapters, various oncogenes that are transcription factors, and discovering that they called so-called oncogenic stress or apoptosis. That's explainable in a model in which this increased bioenergetic demand, not matched with increased nutrient supply, creates the apoptotic cascade that reads out in the BCL2 family. It also argues that if you're going to have an initiating lesion of cancer, if we understand that cancer is a series of genetic lesions, that this is not a very effective lesion to initiate cancer. And that's why so many mouse models that initiate with these kinds of oncogenic lesions take months before you see a tumor to play out. In contrast, we argued, if there was such a thing as fuel signaling, and you had a mutation in the receptors or the signaling that directed that process, this would promote cell survival because the cell would take up, in response to this increased or sustained signal transduction that's oncogenic, increased glucose uptake over its demand and need. That would fuel increased ATP production that would make transcription and translation more effective. The cell would be able to deal with that, but ultimately would create, by overeating, the extra reactive oxygen species as its electron transport chain can no longer, it's now exceeded its capacity for ATP production. And that would promote, as the cell overeats, the extra carbon can be secreted as lactate. The increased ATP-ADP ratio will maintain bioenergetics and therefore cell survival and resistance to apoptosis. And the reactive oxygen species will take this now cell-autonomous for survival cell and begin to mutagenize it through classic Fenton reactions, as those electrons that escape the electron transport chain are turned into hydrogen peroxide, diffuse to the nucleus, in the presence of molecular iron, start to damage bases with hydroxyl radicals or superoxides. And so this is interesting because this predicts that if you were going to try to initiate a tumor, this is the kind of lesion you should initiate with. And so as we were starting to think about that and ask, are there any signaling paradigms that actually control primarily the uptake of nutrients such as glucose? We were reminded by our colleagues in endocrinology that, of course, there was one well-characterized signal transduction pathway that did exactly that. Told cells to take up more glucose than they actually need for themselves, and instructs them to pack those extra glucose away for the rest of the body. That's the insulin signaling paradigm. And insulin-responsive tissues, that's muscle, fat, liver, take up more glucose than they need in response to the activation of receptor tyrosine kinase by its proteinaceous ligand called insulin. That activation of the protein kinase domain leads to the recruitment of adapter proteins that ultimately activate PI3 kinase to convert membrane phosphoinositol into PIP3 that recruits to the membrane in series two serine threonine kinases, PDK1 and AKT. And in insulin-responsive tissues, that's sufficient for AKT in its activated form to direct the cell to place glucose transporters on the cell's surface to capture glucose, phosphorylate that glucose that is captured intracellularly so it can't escape back out the transporters, and commit that glucose to glycolytic metabolism by the indirect activation of PFK1 by the last step described in biochemistry of regulation of bioenergetics. AKT's preferred substrate in this pathway is PFK2, which produces the allosteric regulator and activation of PFK1. And that pathway instructs the cell in response to the sustainment and magnitude of signal transduction to take up glucose in exactly a precise proportion. So the cell's not making the decision, signal transduction is making the decision. We went on to take a variety of non-insulin responsive tissues and showed that if we transformed them with AKT that was constitutively active, it directed all of these processes in every tissue we could find. And we found that in every tissue where their survival receptors had been identified, that they directed this process because they mimicked the insulin receptor, except they were lineage-specific tyrosine kinases that responded to lineage-specific transcription factors to initiate this same signaling paradigm. At the same time here at the NIH, they came to our rescue in the TCGA because as new sequencing was done of human tumors, it was discovered that the most common activating mutation in human cancers after RAS is activating mutations of the catalytic subunit of PI3 kinase. And when you put those mutations found in naturally occurring tumors in human beings into cell lines, they direct exactly this process. And the second most commonly lost tumor suppressor gene in human spontaneous tumors is the negative regulator of this pathway, P10, which is the phosphatase that it inactivates this pathway by degrading PIP3. So, in essence, it appears that in fact this is a pathway known about by cancers. The difference is it's not insulin that's the ligand, it's lineage-specific survival factors. And you can demonstrate this, so I'll just show you very quickly. These are hematopoietic, non-transformed hematopoietic stem cells that are grown optimally in the presence of IL-3, and they'll grow very nicely under those conditions and undergo an exponential growth curve if we passage them in culture. If we put in an AKT transgene, which is expressed at low levels in the absence of induction, but at much higher levels when it's induced, we see in direct response to the introduction of AKT or its induction, there's an increase, corresponding increase in glycolysis. There's no change, however, in the growth rate of these cells. These cells have fully saturated their growth needs already through the IL-3 signal transduction. But at the highest level of glycolysis that can be induced by this level of activated AKT, they actually reproducibly suppress their oxygen consumption, exactly what Otto Warburg described. In pair samples, this is a meaningful reduction in oxygen consumption simply because the high rate of glycolysis is now satisfying the cell's ATP demands. But there's a problem here. If the cell takes up this much glucose in excess of its needs, it's got it someplace to put it, and these are not professional storage tissues. These are hematopoietic progenitor cells, and so to maintain carbon homeostasis, because the cells can't put the carbon into their growth curve, they have to get rid of the extra carbon, and they do that by converting the terminal step of the glycolytic pathway, pyruvate, into lactate and secreting it from the cell. And there's an exact correspondence of the extra carbon that's taken up by glycolysis being committed to secretion by lactate. This is aerobic glycolysis. It's overeating glucose and getting rid of the carbon that you can't effectively use to maintain survival or bioenergetics. And that's what the clinicians here in this hospital have been using to diagnose cancer for now the last decade. In fact, people recognizing Warburg's independently had discovered that, in fact, you could use positron emission tomography to image the high avidity of tumors for glucose that occurs in naturally occurring human tumors. You do that by taking a positron emisser, 18-fluoro-2-deoxyglucose, injecting it into the forearm of a patient, and then asking, by scanning the patient over 15-minute intervals, where does that glucose get taken up in the body. It's a very reassuring thing from what we all learned in organismal physiology. The first tissue that lights up are all the hemispheres of the brain. The liver is the next most avidly recognized in terms of glucose uptake, and then this organ right here is the heart. But at every point of this patient, which has indolent thyroid carcinoma, is imaged, the tissue that takes up the most glucose at every time point is this goitrous adenocarcinoma, or papillary carcinoma rather, of the thyroid. And when this tumor was sequenced, this tumor had an activating mutation in the TSH receptor, which is the lineage-specific tyrosine kinase receptor that initiates this signal transduction pathway that makes the cell scavenge all the glucose that floats by it in the environment. This cell has no proliferative rate because this is an indolent thyroid cancer that's grown over a 7 to 10 year period in this patient's neck. And it's not hypoxic because, in fact, the glucose itself got through the vascular system to this tissue first. And so these are the natural occurring tumors that have activating mutations that actually direct cells to take up extra nutrients, and therefore a push system to put in more nutrients than the cell needs and push the cell to being resistant to apoptosis, pushes the cell to have all the building blocks to engage in macromolecular synthesis. Now, at the time we proposed this then, we said, well, that's for us a pretty cool insight, and allows us to hypothesize the following thing. It suggests to us that many oncogenes, if this is right, that cells lack the cell autonomous ability to take up nutrients, and many oncogenes that we previously had not understood well might actually primarily have evolved to regulate cellular metabolism in multicellular organisms. And that the anti-apoptotic genes and the tumor suppressors might be negative regulators of that pathway so that cells would match their bioenergetic resources to the demands that are being put on them by cell proliferation. And so at the time we hypothesized that, it seemed like a sort of a crazy idea because Bob Weinberg had just finished the single author tree test, the beautiful book I urge the students and postdocs to read, in which he compiled all the known molecules involved in experimental carcinogenesis in this beautiful signaling diagram that's delivered to the cell surface through receptors, adapter proteins to transcription. At the same time, Don Nicholson was retiring from the University of Bath in England, where he had produced, for every one of us that's ever seen a metabolic chart on our cell wall, he's the guy that produced it. This is the 23rd of those charts he produced, and at his retirement party he said, thank God we'll never need to produce another one in that year because we know everything we'll ever need to know about metabolism. As we were hypothesizing that in 2007, however, there was not a single gene on this chart that was on this chart, and vice versa. And that seemed to us impossible, because these are the two most important vital things for the vitality of life. The ability to initiate replication through signal transduction and the biochemistry to enact it. And so we went on to try and ask, what are the advantages of overeating glucose and potentially other nutrients to cells that want to engage in cell-autonomous proliferation, such as cancer cells. So we went first into this process of why is it that aerobic glycolysis is such a special or selected event in tumors. Recognized from the 1920s, it's absolutely recurrently done, it's imaged every day. 70% of human tumors have that glycolytic phenotype as manifest by a PET scan. And the first thing we discovered is that when you engage in glucose metabolism in excess of cell need or demand, it allows the cell to reprogram mitochondrial physiology to the more ancient way in which Metazoan or eukaryotic organisms came to acquire an intercellular pathogen that became the mitochondria. And that is that the initial symbiosis, that's really been shown very effectively now by evolutionary sequencing. The original symbiosis depends not on its ability of the mitochondria to carry out oxidative phosphorylation, but actually to provide unique synthesis events for the host. And the two unique syntheses that seem to occur throughout the entire eukaryotic kingdom are, in fact, the ability to engage in novel lipid synthesis through the mitochondria and to engage in iron-sulfur cluster formation. And so armed with that insight, we went and asked, is there something special about lipid synthesis when cells are overeating glucose? And what we discovered is that the TCA cycle is entirely reprogrammed in the mitochondria under that condition. And cells, as they initiate the production of citrate in the first step of the TCA cycle, couple that production to the excretion of citrate into the cytosol. And there it encounters, citrate encounters another AKT-regulated enzyme complex, ATP Citrate Lyase, and that ATP citrate lyase is responsible for the production of all the acetyl-CoA that's necessary for a cell to engage in net lipid synthesis and in acetylation reactions. We got fooled, we believe, a number of years ago into thinking cells have tons of acetyl-CoA because there's a lot of acetyl-CoA produced during the TCA cycle, but that mitochondrial acetyl-CoA is not accessible to the cell for synthetic reactions that occur in the cytoplasm. And so we were able to go on and show over the next couple of years that this acetyl-CoA is the source of 90% of the synthesis of all the fatty acids used for phospholipid biosynthesis as a cell expands to produce extra lipid bilayers. A large portion of the cholesterol the cell produces in cis, and it's responsible for all the isoprenoid modifications of signaling molecules and other things that allow for specialized localization in things like signaling rash, all the farnesylation, granulation, palmitylation that allows proteins to be modified and aggregated at membranes. In addition, more recently, we've come to appreciate that the dynamic range of acetyl-CoA produced through the rate at which glycolysis flux through this pathway to produce an ACL, acetyl-CoA, is entirely determinant of the level of protein acetylation, particularly in the nucleus. And that had a profound influence not just on thinking about survival and metabolism, but ultimately how cell uses remodeling of chromatin to inform the level of gene transcription in cells. I'm just going to show you two at least ideas about this since it's all been published. It had already been well established at the point that we started our work that declining bioenergetics, because of an increase in the NAD-NADH ratio, was critical for the activation of histone deacetylases of the sirtuin family. SIRT1, which has been so popular on the list as a longevity gene, as an anti-cancer gene. And what sirtuins do then is deacetylate the major histones and drive chromatin into a pattern of organization that makes it less inducive for gene expression, particularly in the form of gene elongation. And what we realized is that the dynamic regulation of production of acetyl-CoA could directly oppose that signal. This would be a low energy signal, low flux in the glycolysis produces this condition. High flux of glycolysis will produce this condition, and we found a tenfold dynamic range of acetyl-CoA in the cytoplasm based on the rate of aerobic glycolysis. And we found that, or we tested first and then found that histone acetyltransferases were highly sensitive in that dynamic range of acetyl-CoA produced by aerobic glycolysis, and particularly the GNAD or GCN5 subfamily of histone acetyltransferases do exactly the opposite reaction under conditions of aerobic glycolysis and will drive chromatin into a place where gene expression is enhanced throughout chromatin network. And that makes sense. As a cell has robust source of energy, it can more faithfully engage in more production of initiated transcripts. And so I'm not going to go through this experiment. We dynamically manipulated mitogenic signaling, so these are resting fibroblasts that have been stimulated with a cytokine cocktail, and we've done that over 4 days in the conditions of a physiologic level of glucose or a diabetic level of glucose in the culture media. And what you can see here is the global acetylation of histone H3 and H4, the greater substrates of acetylation reactions in the histones, is reasonable in the resting cells, but it undergoes a dramatic induction when on mitogen stimulation, and that could be profoundly retarded if we do RNAi against ATP Citrate Lyase. And that's most dramatic when you have both a high extracellular glucose and induction by mitogenesis. You can see a 40-fold induction of acetylation of histone H3 and H4 that's completely dependent on ATP citrate lyase. And so that says that the level of acetylation is controlled on the negative sense by the sirtuin family of genes when you're bioenergetically compromised, and the positive sense by flux through the glycolytic pathway. And so that sits a very beautiful argument in which the generalized state of your ability to engage in transcription versus activation versus repression is controlled by the level of histone acetylation and a variety of marks that others have worked on. So we're just talking about the substrate availability. Sirtuin's deacetylating to silence when NAD dominates because it's not receiving electrons by glycolysis. And the GNAD's dominating when there's a high flux of glucose through the glycolytic pathway. NAD is low and acetyl-CoA is high, which drives acetylation of the histones to increase transcriptional activation. And we and others have been able to show that a preferential site for this is a positive feed-forward loop on the genes that commit the cell to glycolytic metabolism. It's a positive feed-forward loop that locks the cell into dependence on glucose. And that's then sort of explains why it is that in both cancer and in many model organisms that have been, all model organisms in fact that have been studied, signal transduction through the PI3 kinase AKT pathway and then secondarily through TOR, through AKT's direct and indirect ability to activate TOR, is the controller of cell size. Because it allows cells, it directs cells without any transcriptional intervention to reprogram all of cellular metabolism. To capture glucose extracellularly, committed to the glycolytic metabolism in sufficient quantities that citrate can be produced in excess to produce non-essential amino acids, which I haven't talked about, but lipids for lipid biosynthesis. And TOR's activation reprograms amino acids away from catabolism in the TCA cycle and into tRNA charging and increased protein translational initiation, and therefore increased protein synthesis. So under all physiologic conditions in all model organisms that have been studied, spontaneous activation of this signaling pathway that I've just talked about makes for a larger cell at all stages of the cell cycle because a cell is simply a bag of proteins surrounded by a lipid bilayer. And secondarily, there's positive information of those two pathways that I said were regulated independently in the sort of the modeling that we started with, because citrate informs through its ability to modify acetylation increased ability to efficiently transcribe genes, and TOR increases the ability to officially activate translation, particularly a translation initiation. So you inform the two remaining processes required for growth, protein, gene transcription and protein translation. And so we were pretty happy with this as an explanation of, in fact, what is the fundamental basis of cell survival, the absence of the ability to take up sufficient carbon to survive, and the reprogramming of that carbon directed at an oncogenic step. And a few years ago, we started to think about this, and maybe this could inform more about cancer than just glucose metabolism. And we were informed by that because as a medical oncologist, although I'm very interested in the mutations in cancer and these things, and this aerobic glycolysis that can be imaged by PET scan imaging, I've never had a patient die of one big cell. In fact, a cell has to be able to divide to produce the kind of lesion we associate with the pathogenesis of cancer. It's the expanding clone of cells and their ability to acquire new properties and the ability to metastasis, and particularly in human patients, that was important. And you cannot build all the building blocks necessary for cell division simply through these pathways. And what one needs is a robust source of reduced carbon to build nucleic acids that was missing from this cascade. So we went looking for whether there was a pathway dedicated to the uptake of reduced nitrogen that would allow cells to inform nucleotide biosynthesis and the remaining synthetic reactions that we hadn't yet put on this cascade of growth.
And to make a long story short, in hematopoietic tissues, as I've showed you, receptor tyrosine kinases can initiate this pathway on this side of the arm, but we discovered that cytokines, particularly dedicated to the JAK-STAT signaling pathway, informed the transcription of the essential factor that so far we and others have found that regulates nitrogen uptake. And that nitrogen uptake is directed by Myc as a transcription factor, which puts in a cascade of genes that controls the uptake and metabolism of glutamine, much the way this signaling pathway controls the uptake and metabolism of glucose. And the uptake of glutamine provides a nitrogen source for de novo purine and pyrimidine biosynthesis. It provides a carbon source to allow glucose to be redirected into ribose biosynthesis, and it allows a substrate that can also be converted, if metabolized in excess, to produce NADPH to fuel the synthesis of nucleotides and lipids. And so that seemed to complete all the major building blocks that we'd need for cell growth, and consistent with that, Anton Burns made a mouse that in the immunologic lineage, the B cell lineage, has the activation by an immunologic enhancer of AKT and Myc at day 18 of embryogenesis, and instead of now taking months, these two individual mice, this mouse, a Myc B cell mouse, will produce tumors around 6 months of age, an AKT mouse about 9 months of age. If he bred those two together, as Anton produced, because he found that these two genes complemented each other, he found that the first B cells are made in a mouse at day 18 of embryogenesis, and every animal dies of its tumor at birth 3 days later. Because the minute the cell commits to the B cell lineage, these two pathways are fully on, and the cell has a cell cycle time of 5 hours. It doesn't think. It simply divides. And it divides wherever it is, no matter what else it's doing. So what was interesting about that is we thought that the Myc pathway would primarily be telling us about nitrogen metabolism. And then we did this experiment, which got us totally confused. In complete media, tissue culture cells, now for the students and postdocs, the reason we knew glutamine would be an important part of this is the dirty little secret of cell culture is that we add 10-fold more excess of glutamine to every tissue culture media in the world. Since we're an immunology lab, we double that because we don't trust anybody else. All the immunologists that are here do that, so don't worry, we're not unique. And so we thought the glutamine would be special. So we demonstrated that if you take Myc-transformed cells, and we can either find Myc-transformed naturally occurring tumors or make Myc-transformed cells, and what we found is they were addicted to glutamine. If we took glutamine out of the media, left the glucose at 11 millimolar, as it was in this tissue culture media, kept all the other nutrients present, but just took glutamine out, every cell died within 3 to 4 days in the tissue culture media. The surprise to us came when we tried to ask what would rescue this death, and we found the only thing that was necessary to rescue the cell death was the carbon backbone of glutamine, alpha-ketoglutarate. We didn't need any of the nitrogen to survive the rest. And we did need some of the nitrogen to initiate full proliferation, but no cell dies if we just added alpha-ketoglutarate. And that was a puzzle and made us want to dig deeper into this. Now, what we learned from those studies is that, in fact, for robust growth, to be able to fuel the citrate biosynthesis that I talked about, as citrate is exported, you don't only export the carbon equivalents that come from pyruvate that I talked about. Remember, pyruvate's converted to acetyl-CoA. It's then condensed with oxaloacetate to produce citrate. And when it's exported and used for synthesis and various synthetic reactions downstream of this, you need a source of something to produce extra malate that then re-enters the TCA cycle as an anapleurotic substrate, something to replenish the TCA cycle to produce oxaloacetate. And we discovered that in effectively growing cancer cells that were transformed with Myc, all of the anapleurotic substrate that maintains the TCA cycle enters as alpha-ketoglutarate derived from deamination of glutamine. And so the rescue of cell survival we were seeing was the ability of alpha-ketoglutarate to build up a pool of the cell that maintained the integrity of the TCA cycle, or that was a sufficient explanation. In fact, in Myc-transformed cells, we find that this alpha-ketoglutarate pool, which is in equilibrium with glutamate to exchange nitrogen, is about 1/2 to 1 millimolar. And that's a unique pool. We never see glutamate alpha-ketoglutarate levels combined build up to those kind of levels, except when the cells are instructed to scavenge glutamine from their environment. And so we asked, what's special about having this level of alpha-ketoglutarate? And that'll be relevant to the last 15 minutes of my talk. One thing that came out of that study, and it's been published by 6 groups in the last 6 months now, including ourselves, is that glutamine metabolism is special when cells want to survive hypoxia. When you want to survive hypoxia and you want to generate ATP, I think as everyone knows, you engage in glycolytic metabolism, as I showed you in the first slide. That means there's no substrates to supply the TCA cycle, and therefore a carbon to do synthetic reactions to build growth. Yet when you cut yourself and you have a wound, and you need to grow either the vasculature back or recover the integrity of that tissue, in the absence of molecular oxygen, you need some way to build the lipids and the non-essential amino acids under those conditions of hypoxia. And so with that kind of reasoning, 6 laboratories literally simultaneously asked, I wonder if this signal transduction through glutamine could do that. And what we and 2 other laboratories described earlier in this year was that in fact glutamine metabolism downstream of Myc could fully inform cell growth under hypoxia. Glucose metabolism would provide the ATP, but glutamine, now by providing this large pool of alpha-ketoglutarate in the TCA cycle, even though there's no molecular oxygen, so you can't move metabolism in this direction, because that would require an electron acceptor and oxygen to do electron transport, alpha-ketoglutarate can now work in reverse as the TCA cycle is reprogrammed, be carboxylated, so this is reductive carboxylation, into citrate, a disocitrate, which is then by aconitase converted to citrate and exported to provide the same synthetic fuel for both acetylation reactions and lipid biosynthesis that I talked about through glucose. And a cell can grow using glutamine as a substrate to protect the mitochondria under hypoxia by preventing redox stress in the mitochondria, and at the same time provide synthetic fuel for glucose. So this pool of alpha-ketoglutarate in response to glutamine provided a special adaptive ability to cells that had lesions that scavenged glutamine from the environment. And that has been extended now by several laboratories. As we were describing this and getting quite satisfied with ourselves, that this was a pretty cool pathway downstream of the Myc oncogene that had the components of the transporters that capture glutamine, the deaminated glutaminase, the either transaminated DNA, which are GDH and ALT, entered into a partial TCA cycle. We found that the NADPH was produced by malic enzyme converting malate to pyruvate, producing NADPH, which provides the electrons for the synthesis reactions required for nucleotide biosynthesis and lipid biosynthesis. And you could throw away any extra carbon that the cell didn't need or hold on to by using LDHA, another Myc target, out into the cytosol. And what was exciting about this for us, and shows how nerdy we are, is that when we add up cell growth as a problem, the limiting substrate for us is the lack of any known way to produce enough NADPH to actually fuel cell growth. You cannot do lipid synthesis, you cannot do nucleotide synthesis without a robust pool of NADPH. If you go and ask your test book, I think none of you know any synthetic pathway to produce NADPH, except for a few of you who remembered your biochemistry well enough because you got the honors, that enzyme is G6PD, which is the pentose oxidative arm of the pentose phosphate shunt. And we knew early on in these studies that that wasn't the possible answer for when NADPH goes for growth, because there are 3 million men walking around the Mediterranean basin that have no G6PD. And they got through embryogenesis without G6PD, and they have been well studied in all of the Mediterranean countries. They have identical rates of cancer to their age-matched controls. So G6PD is not the substrate that produces NADPH for growth. And so there were only 2 remaining candidates, and we were very excited about malic enzyme, because malic enzyme can produce from malate and pyruvate in this pathway NADPH. In fact, Patrick Ward in the laboratory went on to show that Myc-induced malic enzyme could produce NADPH, but when we did that, we discovered we got one thing about this model wrong. The malic enzyme produced by Myc is actually it has a mitochondrial localization sequence is actually in here. And so even though it was true that Myc-induced NADPH production, it was in exactly the wrong place, because you produce NADH and NADPH in either the cytosol or the mitochondria, and they're independent pools that don't exchange. And so now we had a problem, because although there are well-described ways to convert NADH from the cytosol to the mitochondria and back, there were no known ways to transport NADPH across the membrane. And so Patrick Ward in the laboratory a couple of years ago recognized that there were 2 other additional enzymes related to the TCA cycle enzyme isocitrate dehydrogenase that I just talked about that controls things. They're called IDH1 and IDH2, and they are NADPH-dependent enzymes, and they are responsible for the reductive, ADH2 is responsible for the reductive carboxylation I just talked about. It's also responsible, at least in the data we've been able to derive so far, to be able, in this reductive carboxylation, export into the cytosol and then reverse metabolism through its homolog IDH1 and the cytosol to regenerate alpha-ketoglutarate to transfer one NADH equivalent from the mitochondria to the cytosol. And so Patrick built up a considerable amount of data that in MYC-transformed cells, the way cells deal with their NADPH problem is to produce it in the mitochondria and then export it through this shuttling pathway. And in support of that argument, he was able to show that if you eliminate either IDH1 or IDH2 by RNAi from MYC-transformed tumor cells, they don't die, but they lose their ability to proliferate in tissue culture compared to the unmanipulated, un-RNAi-treated cell. And so we were incredibly excited to sort of say, this is a really cool thing. This is a new pathway of metabolism in which we equalize electrons away from the compartment where normally they're used to fuel oxidative phosphorylation. We transfer them back to the mitochondria to fuel synthetic reactions, when the following discovery was made by Bert Vogelstein and his colleagues at Hopkins. It's been extended now by many groups. In fact, this slide is not even up to date. Bert was able to discover in secondary glioblastomas the pathognomonic lesion of isocitrate dehydrogenase. Essentially, every secondary glioblastoma, that's a glioblastoma that arises in a bed of an indolent glioma, initiates with a lesion in either the cytosolic form of IDH1 or the mitochondrial form of IDH2. It's now well over 90% of all intermediate grade gliomas have mutations in one or the other gene, and they appear to be the initiating gene. Subsequently, it's been shown that 60% of chondrosarcomas have this as an initiating lesion, 30% of adult AMLs, and more recently, a large percentage, over half of cholangiocarcinomas have IDH1 or IDH mutations. And that didn't fit our data, because when Bert characterized them, these enzymes had completely lost their ability to convert isocitrate to alpha-ketoglutarate. And I had just argued that that ability was required for the shuttling of NADPH equivalents to be able to effectively provide the energy substrates for cell growth. But there was some hope for what we were thinking and what they were thinking, because what they had noticed is that all mutations were missense, and that all the missense mutations occurred in a single arginine in IDH1 and its identical arginine in the reaction center on IDH2. And in every case, one IDH1 allele or one IDH2 allele is always maintained wild type. So there's always wild type activity in the other homolog. And so if you actually just look at that reaction, it starts to explain what they were finding by these recurrent, naturally occurring human mutations now in 4 major malignancies and soon to be a 5th. And that is, this is the reaction product. These are the amino acids that line the substrate binding site of isocitrate dehydrogenase. Here's isocitrate modeled into that site and how it's coordinated by bonds with the enzymes in the IDH gene. And what you can see is that this blue carbon, which we've labeled here because it's the CO2 that leaves as isocitrate is decarboxylated into alpha-ketoglutarate, is its ability to be bound into the site is coordinated by the arginine 132 in IDH1 and arginine 172 in IDH2. And these were the mutations, and the only mutations that Bert Vogelstein's lab was able to find in gliomas. That makes sense then. If you can't coordinate the thing that you're going to break off of this molecule, you can't bind isocitrate very effectively. But it's interesting because the product of the reaction, the normal forward reaction, should bind absolutely with wild-type affinity because it doesn't have this CO2 group. And so armed with that information, Josh Rabinowitz, with the investigators at IDH Pharmaceuticals, transformed normal cells with an IDH1 or an IDH1 mutant and asked, is there any new metabolites that are produced? And what he discovered is a new metabolite based on metabolomics analysis, and it reads out in the following way. It makes perfect sense. The metabolite is 2-hydroxyglutarate, and it works because alpha-ketoglutarate can bind to the reaction pocket with wild-type affinity because it's not dependent on that arginine for its binding. But it is absolutely dependent on that arginine that's been lost for the ability to engage in reverse carboxylation, to be able to accept a CO2. Without the arginine to coordinate the readdition of that CO2, that part of the reverse reaction can't occur, but it can still accept electrons from NADPH, except now the product is a hydroxylation of the beta carbon. And so you produce 2-hydroxyglutarate instead of isocitrate. Now what has been going on by a number, and so then you go look at tumors that have mutations in IDH, and what you find is that those tumors, when isolated, this is just some data sets that we derived from AML patients, tumors that have wild-type IDH that are AML have either no detectable IDH, or this is the level of detection of a GC mass spec. In contrast, these mutations that were also seen in the leukemias, IDH132 and IDH2R172, build up, this is a log scale, 10 millimolar intercellular concentrations of 2-hydroxyglutarate. That makes it the most common metabolite inside the cell. We then screened all leukemias just for 2-HG and discovered, in fact, another group of mutations that had high 2-HG, went and sequenced their enzymes and discovered that they had a mutation in IDH2, but now it was in a different arginine, 140, and that sort of sealed the deal for us because 140 is the other amino acid in the reaction site that coordinates that CO2 looping group. And so what that says, and actually what's been now shown by the investigators at GSK that have been investigating this, is that the preferential operative moiety inside the cell is actually a heterodimer of the mutant enzyme with the wild-type enzyme. The wild-type partner converts isocitrate in the presence of NADP as an electron acceptor into alpha-ketoglutarate and CO2, passing electrons to NADPH. And the concentration of NADPH and alpha-ketoglutarate at this reaction site then drives the production of the reverse product, 2-hydroxyglutarate at the mutant. And so this is a heterodimer that's naturally occurring as a result of this mutation, has a neomorphic activity that converts isocitrate to HG, and this is just a factory to do that. And so the cell slowly but surely builds up a steady state level that can be measured in millimolar of 2-HG. So that became interesting because this isn't a genetic lesion that's building up. It's a metabolite. And so the question is, how does a metabolite influence anything that has to do with cell biology? And so we took advantage of the fact that in leukemia, we were surprised to find a mutation that had a penetrance of 30% in our patients because it made no sense. We thought we knew every leukemic mutation that existed based on sequencing. And so we asked, because we knew so much about leukemogenesis, were there any genes that were always mutated when IDH was mutated, or were there any genes that were never mutated when IDH mutated, to suggest that they either depended on each other for their functionality or they were epistatic to each other. And what we discovered is there was one gene that was epistatic with IDH1 and IDH2. When you look for patients, and this is just a connection gram of patients' tumors that had more than one mutation, these are all the tumors that had IDH1 or IDH2 mutations. They could have mutations in any other known oncogene that caused leukemia except one, and that was TET2. And in reverse, patients that had TET2 mutations could have mutations in any other known oncogene except for IDH1 or IDH2. Highly statistically significant and replicated through multiple databases. And that gave us at least the clue for what has been the initiation of this second connection of metabolism to epigenetics. And that was the realization by Angela Rao's laboratory that TET2 is an enzyme that can initiate the demethylation of DNA at CpG islands because TET2 takes alpha-ketoglutarate as substrate, strips electrons out of it, producing succinate, and chances of electrons in a hydroxylation reaction that initiates the destabilization of methyl C. And that initiates ultimately either actively or passively DNA demethylation. And 2-hydroxyglutarate, because it is a homolog of alpha-ketoglutarate, looked to us like to be a good competitive substrate that would block this reaction. So we went on to test that idea. We adopted an assay that Angela had described. You transfect in TET2. You can see the cells, because they have a flag tag, that are TET2 transfected, and those cells acquire huge hydroxylation marks on their DNA as the exogenous production of TET2. And if you co-transfect in the mutant IDH1, you completely block the ability of TET2 to induce hydroxylated DNA. But wild-type enzyme does not do that. And you can see that's a 1-log effect if you do it by FACS analysis. And we went on then to sort of say that this is something I don't have any time to show any of the data for. We now believe that that burst that comes upstream of Myc that produces this pool of alpha-ketoglutarate is absolutely essential as the cofactor that allows demethylases of both DNA and histones to activate the return of silenced chromatin from an inactive state to an active state. And on that basis, if you can't reactivate genes that are silenced to allow cells to engage a differentiation program, they're stuck in their current or stem cell-like state. And 2-hydroxyglutarate, by acting as a competitive antagonist, prevents the cell from seeing or utilizing that alpha-ketoglutarate burst of energy to initiate demethylation of silenced chromatin. And so to prove that, we demonstrated, I'm not going to, because I'm going over in time, when you either transfect in mutant IDH or you delete TET2 by shRNA, you get persistence in cell culture and hematopoietic stem culture of c-Ket positive stem cells. The cells lose their ability to differentiate, and that fits a model of what we already knew from the very beginning about AML. It's lost the ability to differentiate, and that the loss of either TET2 to initiate the opening of chromatin or 2-HG to competitively inhibit alpha-ketoglutarate to be used as a substrate by the TET enzymes completely blocks cellular differentiation and therefore initiates the block to differentiate that contributes to AML pathogenesis. The question is, what about gliomas? Because at the same time we were doing this work, we were also looking at gliomas. We didn't see any TET mutations, but in doing those studies, we realized that there's a different set of methylation marks that also depend on this alpha-ketoglutarate burst downstream of Myc for their demethylation, and that's the histone repressive marks on H3K9 and H3K27, which are required to maintain silenced chromatin along with methylated CpG. We were able to demonstrate, or others had demonstrated, that the class of enzymes that do those demethylation reactions of the repressive marks are the Jumanji C histone lysine demethylases, and they depend on exactly the same enzyme kinetics, alpha-ketoglutarate being converted to succinate, and we were able to demonstrate if you take the H3K9 demethylase KDM4C, you add alpha-ketoglutarate or 2-HG as just shown here, you take histones that are heavily modified on H3K9 that's naturally occurring, you add them to culture, add the enzyme, in the presence of 1 millimolar alpha-ketoglutarate, you completely demethylate them. Add 2-HG and you completely block that demethylation, but it's competitive. As we dose up alpha-ketoglutarate, you reverse the ability of 2-HG to convert. If you go then into spontaneous human gliomas, what you find is H3K9 is highly methylated in the oligodendrogliomas associated with IDH1 and IDH2. This is just immunohistochemistry to demonstrate that. Mutant ones have high H3K9 methylation, and that's true and highly statistically significant in IDH2 mutant tumors. And that when we take an astrocyte primary cell line and introduce a mutant IDH, we actually first see histone methylation, and that's subsequently followed several generations later by DNA demethylation. It's not affecting the TETs. It seems to be cooperativity of the inability of to demethylate histones impairs the ability to demethylate DNA. And based on that, independently of our work, the group at USC that works on epigenetics was also looking at gliomas and asking for glioblastomas across the board, can we define subgroups based on their methylator phenotype? They defined a group, red is heavily methylated in this group, and you can see that there's a group over here on this side that is heavily methylated by their analysis compared to all other glioblastomas. This makes up 10% of the group. And when you ask by gene expression phenotype, those are the pro-neural tumors exclusively, the ones that are stuck at the true stem cell state of glioblastoma. They're highly hypermethylated, and when one then analyzes for genotype, you find that they're all mutant for IDH1, and all the mutant IDH1s are in this group. And so in an independent test in a separate tumor, the interference with metabolism by this 2-HG completely blocks the ability to access new genes by demethylation. So we're stuck with a model that histone and DNA methylation requires active intervention through a metabolite substrate, which is alpha-ketoglutarate, to be provided as a substrate for the exquisitely specific enzymes that then demethylate sites to activate repressed chromatin to allow cells to differentiate. And when that's blocked by 2-hydroxyglutarate as a global inhibitor, the cell is stuck in maintaining the stem cell state it has. And so at that point, we think that this is very informative in terms of a balance that epigenetics is really creating through metabolism to instruct what part and how much of the genome should we access access based on the nutrients that the host is making available to the cell type. And mutations in that pathway, either by oncometabolites, by signal transduction genes, or by metabolism, can inform this process. So that's all I have to say. I apologize for going a couple of minutes over, but I'm happy to stay and take any questions, and I really appreciate your attention. I guess I was going to just show you that if you actually do this, you completely block neural differentiation. And I'm just going to show that as a final point. But if you take primary, this has just been published, if you take primary cells, introduce an IDH mutation, and then ask neurospheres to differentiate, primary cells lose their complete ability to differentiate, which is what that last slide does. Overexpression of wild-type IDH has no effect on that process. So this seems to be a critical connection between metabolism and the ability of cells to engage in new programs by accessing new DNA through methylation. So thank you very much, and I'm happy to take any questions. Somebody has to be brave and be the first one, or we could all go home. Yeah. If you go up to the mic, because there's people outside of this room, apparently.
Thanks for a great talk. I know you are talking about cancer here, but since you are talking about you have a way to block differentiation, do you think you can do something with stem cell therapy here? Because we know like it's kind of annoying, you'll get stem cells and they will differentiate to some other cell types.
So the surprise is, which doesn't really answer the question, could we use this kind of information to maintain stem cells or do something with stem cells? That's the question. The only thing we can do to get cells to really show us their true core phenotype in a state in which they will never differentiate is if we starve them for the two nutrients that I just told you about. If we starve cells for glucose and glutamine, if they don't commit suicide, they retract to maintaining their pure phenotype. And then even though we know they're capable of differentiation, we can't add a growth factor that'll make them do it. In the absence of metabolism, in the absence of these two intracellular pools of metabolism, we cannot force a cell to differentiate. It's as if a cell needs to be told through these medical pathways, it's now time and you have the resources to take this leap of faith to go be something else. And it is never done under a low energy condition. So the problem is starving a cell isn't a very good way to maintain things or to get more stem cells. So it's not it was an interesting, we liked the idea, we did it, but it didn't work. And so we do think that what really this is saying, and I didn't have time to talk about, if you look at another part of what Peter Laird just published, there is another group of cells that are stuck in the stem cell state. They haven't been talked a lot about. They're the ones that are completely hypomethylated at both the DNA and the histone level. And I think that really means, and what we're trying to push as an idea to think about, is that if you're stuck with all your genes except the core genes that maintain you what you are, stuck in a heterochromatin state and you can't access them, you're stuck in stuck with the differentiation you have. If on the opposite side everything's open, stem cell maintenance genes seem to dominate and the cell can't differentiate under those conditions, which means, like everything else, there's a couple of barriers to regulating differentiation. You both have to silence stem cell maintenance genes. That's not surprising given iPS cell biology. You have to silence those genes that maintain stemness. But at the same time, you have to gain access to the genes that allow you to be the next cell in your lineage. And it's the dysregulation of that if you now take that model, you can see that almost every cancer type has a dysregulation in one direction or another by this.
Beautiful
Mike.
talk, beautiful odyssey through cancer metabolomics. The question I had, though, is because these pathways are so fundamental, does that pose a problem for an oncologist trying to think of therapeutic interventions? Because you risk disrupting things that are going to affect a lot of different cells in the body.
So there's a lot of long answers to my question, but it's really important, right? Core metabolism is core metabolism. It goes back from prokaryotes to single-cell eukaryotes to all multicellular organisms. We have a common core metabolism, so if you intervene with that, it's not, you might kill the cancer cell, but you're going to kill a lot of normal cells at the same time. You're going to have more toxicity than traditional chemotherapeutic agents. The first answer we have for that we've published in multiple forms is if you go back and ask how do traditional chemotherapeutics that actually cure you of cancer, or the doctor uses and cures a significant patient, almost all of them are in fact intervening in metabolic pathways as their fundamental basis. So anti-metabolites, methotrexate, other things, we went on to show that traditional alkylating agents impair glycolytic metabolism because they consume all the NAD. And so we do actually think that our success in cancer chemotherapy has not been realized as mostly a metabolic strategy. And that tumor cells, once addicted through activation of signaling pathways to reprogram their metabolism to be addicted to these metabolites, are fundamentally different than normal cells that when they lose glucose and they don't have a driving mutation through the PI3 kinase pathway, just say, oh, okay, well, there's no more glucose right now, but I can metabolize fatty acids. And when AKT is constitutively on, it says, you don't get to do that. We need them for growth. So your survival strategy doesn't work here. And we went on to describe a whole tumor suppressor network that's needed for reactivation of beta oxidation and when you lose glucose. So you can do really, for the students and postdocs, really stupid experiments, which is what we do right now. You just take out a single metabolite and ask how does a tumor cell versus a normal cell adapt. And that's how we have categorized, but I didn't talk about any of the tumor suppressors. About half of tumor suppressors fit into a model of being able to accommodate either glutamine withdrawal or glucose withdrawal. And that normal cells activate those immediately, and oncogenic lesions prevent those pathways, or the loss of those genes as sensors lose that pathway. So I think we're going to be able to exploit it. And that gets to the final thing, and maybe this should be the final thing since I've gone over. I think there's a fundamental divide we have in oncology right now. We are very impressed. I'm very impressed. We should all be very impressed. We've discovered oncogenic driver mutations in many human tumors. And we have developed the ability to develop inhibitors of those driver mutations that will turn those genes off. But the real feature is, although we had a tremendous success in Gleevec, most of the time turning those off hasn't really helped the patients for very long. Hasn't cured many people. Because in fact, cancer has an armamentarium of things to get around the inhibition of driving oncogene. And it's hard pharmacologically to completely inhibit an activated oncogene, even when we try and do that. In contrast, all the therapies that cure patients have left the oncogenes in place, allow them to force the cell into doing something that a normal cell equivalent might not do under those conditions, and make that condition harmful for the cell. The attempt to try and divide when there are no nucleotides is a really bad idea. And so that's the basis of most anti-metabolite therapy through the antipurines and antipyrimidines. And so that's the basis of methotrexate. So I think we've forgotten that approach, and we think metabolism will reactivate that approach. The understanding that cancer pathways, although they do all the other things, and I'm not trying for any of the students and postdocs to negate any of the importance of the signal transduction pathways, the genes necessary to orderly progress you through a cell cycle, the checkpoints that we've all talked about. But there has to be fuel to do that. So if you ask a cell to go through a cell division and you sweep out a fuel from it, it's in a lot more trouble than a cell that's just hanging out there saying, I'm just supposed to occupy space and keep alive. It has a lot more adaptive mechanisms. And so I think there are going to be ways to exploit it, but it's not by eliminating the oncogenes, it's by leaving them in place and making what they force the cell to do toxic under changing metabolic conditions. And that's the approach we're exploiting right now. Anyway, thank you very much for all your time and attention, and thanks a lot.
So let me remind you that we have a reception which is sponsored by the FAS in the library, and an opportunity to ask Dr. Thompson the really burning questions that you have. Thank you.